Hello and welcome to today's video lesson. It is on cellular transport and cell size. And the learning goals for today, there's a few. Firstly, is to explain how the cell membrane maintains relatively stable internal conditions via active transport. And that means going against the concentration gradient. Secondly, to understand that endocytosis is a form of transport that usually moves large molecules that can't just diffuse through the membrane. To thirdly, recognize that phagocytosis is a form of endocytosis. To predict the direction of movement when we look at various factors that affect transport and movement, such as concentration, physical, and the chemical nature of materials. And lastly, to explain how the size of a cell is limited by the relationship between surface area to volume ratio. So here you can pause the video and just summarize what you see in front of you. Cool, so let's move on. First up, active transport, and this is from 1.3 of your text, and we have a couple of key terms just down the side there that you can add to your glossary when you're ready. But let's get into it. So the learning goal we said was to explain active transport against the concentration gradient. And what does explain mean according to QCAA? It means to elaborate on something by providing more detail, as opposed to just defining or describing. So last week we spoke about passive transport. That is the movement of particles across the membrane from an area of high to low concentration. And like a ball rolling downhill, we said that it doesn't require any energy from getting from the high point to the low point, from high concentration to low concentration. The other type of transport that takes place in our cells, though, is called active transport. And we said that active transport does require energy. The most famous of all examples of active transport would have to be the sodium-potassium pump. You'll see this in a lot of textbooks. What it does is it moves sodium from outside of the cell and it moves potassium to inside the cell. So here you can see the extracellular space where there's a lot of sodium already, and then over here we can see inside the cell there is a high concentration of potassium. And what happens is essentially we just move sodium against the concentration gradient to where there is also already a lot, and potassium in the same way to inside the cell. So if you look at the diagram, you can see there is already more sodium outside where we are moving it. And so to transport material across or against its concentration gradient, we have to use energy. And energy in the body in cells is in the form of what is called ATP. That stands for adenosine triphosphate. It attaches to the pump, and as it does this, it causes a change in shape, moving the ions and causing their release. At the same time as this, as sodium goes out, potassium also comes in, and that's why it's called this exchange pump. It's this protein that changes shape when energy binds to it, causing work to happen. So lots of cells in our body use this pump to keep the correct balance of sodium out and potassium in to maintain a stable internal environment. It requires 1 ATP to move every 3 sodium ions and 2 potassium ions. And that is an example of active transport. Next up, from 1.4, we're talking about movement of large molecules. So the learning goal was to understand that endocytosis is a form of active transport that moves often large polar molecules that can't just pass through the hydrophobic cell membrane and into the cell. But to also recognize that phagocytosis is also a form of endocytosis. So to understand means to perceive what is meant by something, to grasp it, to be familiar with it, to construct meaning from it. And to recognize means to identify or to recall particular features from knowledge. So if we are to get into it, big scale movement across a membrane is called endocytosis and exocytosis, as opposed to what we saw before, which was just like small little potassium and sodium ions moving in and out. So endo, as the word suggests, like an endoskeleton, means inside. So endocytosis means moving cells inside, from outside to inside the cell. Moving large molecules, I should say. And so here is an image of a phagocyte. If you're wondering, like, where does this actually happen in the body? This phagocyte that you can see here is a white blood cell, and it moves around in our body, in our blood, and essentially eats invading cells. So this phagocyte image you see, it is reaching out and engulfing, it's grabbing, like with these tentacles, all these little bacteria, these foreign cells within the body, to destroy them as part of our immune system. And the way it does this is it actually pulls it in, and then it creates these folds in its membrane. That's what this section is. It engulfs it, and then once it's engulfed, it creates this vesicle surrounding it, and it breaks it down. So that's an example of endocytosis, and it requires energy to be able to move the membrane and do things like that. So it's, of course, an example of active transport. The opposite of that is exocytosis, and exo means outside, and basically it's the exact same thing but in the reverse process. So it's the way of moving large molecules from inside to outside the cell, and it does this by creating this vesicle, and then it fuses with the membrane, and then from there it releases those large molecules into the extracellular fluid. And this might be broken down waste that it's excreting. It could also be the process of excreting like hormones or enzymes or proteins or antibodies from inside the cell to the outside. And here is an image taken from an electron microscope of this playing out in real life. So you can see the membrane, and then there's these large molecules, and the membrane folds in, creating this vesicle, and now it's inside the cell. So that is an example happening in real life. So to summarize, if you're not adding energy, it's called passive transport. But if you are adding energy, it's called active transport. And both of these methods are ways of moving materials across the cell membrane. Then we have endocytosis and exocytosis. And this is the movement of large materials whereby the membrane folds and creates these little compartments called vesicles that either transport material from the outside in or from the inside out. And this allows material that is either polar, meaning it's got a charge, or is too large, or is hydrophobic, to be able to pass through the membrane, whether that's inside or going outside. The last point I want to make, though, to tick off this learning goal, is to mention that phagocytosis is also a form of endocytosis. And phago means like solid food. And it's just this specific term given to when it is a solid material that is entering into the cell as opposed to a liquid, such as a hormone. Then it has its own term, and it's been described as like the way in which a cell eats. It ingests solid particles. We give it its own term, phagocytosis. And you can read more about that on page 18 in your textbooks. Here we have a few key terms down the side that you can add to your glossary as well. But moving on to 1.5, where we talk about direction of movement. And so the learning goal says to predict the direction of movement of materials across cell membranes based on factors such as concentration, physical and chemical nature of the materials. So to predict means to give an expected result of something, to suggest what may happen based on available information, and we will do more on this in class, but here's a snapshot of how these kind of factors play into the direction of movement. And we've actually spoken about a few of these already. So whether or not a substance moves across a membrane depends on a few different factors. That can be concentration, or it can be physical and chemical factors. So this diagram here is taken from page 20 of the text, and it summarizes how the relative permeability allows some things to pass through, but not others. And in this, we can see that small uncharged polar molecules such as water, ethanol, and glycerol can easily just diffuse through this membrane, this hydrophobic region, because it's small and uncharged. But larger uncharged polar molecules cannot pass through this hydrophobic zone. That includes nucleotides, glucose, and amino acids. And so we spoke about the proteins, the role of proteins in facilitated diffusion to allow glucose to pass through. Then we have charged ions such as sodium, such as potassium, and again we have proteins such as the sodium-potassium exchange pump that can allow these ions to pass through, but they can't diffuse by themselves. Unlike small hydrophobic molecules such as oxygen and carbon dioxide and nitrogen, which can easily pass through. So the physical properties we just mentioned are, of course, size, the chemical properties being charge, and concentration allows things to diffuse through easily, such as there's a lower concentration of oxygen inside the cell because that's what our cells use to produce energy, whereas there's oxygen flowing outside of the cells where our capillaries bring blood supply to our cells, and in that blood supply there's oxygen. So there's a concentration gradient whereby oxygen will diffuse from high to low. And it's the same process with carbon dioxide, but the reverse, where our cells produce carbon dioxide, and so we want to get that out, and so there's lots of it in the cell, so it's going to flow outside of the cell naturally, where our blood will then pick it up and take it to our lungs, where we will then breathe that carbon dioxide out. So super interesting, pretty amazing body, hey. So that's that. That is 1.5, and you can study that a bit more in your textbook. Moving on to the last topic, and it is 1.6, where it refers to size of cells and this concept called surface area to volume ratio. And so this again is a bit of an overview, and there's plenty of other awesome YouTube videos out there and more you can read in your text. So the question that I want to start with is this: why are cells so small? We know that cells make up every living thing, and we believe that there's about 37 trillion cells, that's a million in our body. So there's obviously a lot of them, which opens up the question, why are they so small? The answer to that question comes down to this concept, surface area to volume ratio. Before we go too into it though, let me introduce this man after the learning goal. Surface area to volume ratio, we want to explain what that is, and to explain means to describe in details. This is the man. His name is Joel Allen, and he was an American zoologist who back in 1877 invented Allen's rule, named after him. And Allen's rule applies to all endotherms or warm-blooded animals, and it says basically the further you live from the equator, the more you will look like a sphere. And if you look at early pictures of Eskimo families or Inuit families, you'll find they have shorter arms, stockier legs, rounder appearances. The reason why is just like a polar bear. They want to maximize their volume and to minimize their surface area. That is how much of their outer body, say their skin, is in contact with the cold air, because they don't want to lose a lot of energy in the form of heat. But Allen's rule says as you move towards the equator, people are going to look taller. That is, they're more skinny, and it's because they want to maximize their surface area to volume ratio. They want to get rid of heat easily. And this photo is a photo of the Maasai warriors of Kenya doing their cool high jumping dance. So this gets to the idea of why cells are so small. By making them smaller and smaller, we can maximize their surface area to their volume ratio. Another analogy is if we're trying to start a fire and we have two options. We have one really large piece of wood and then lots of smaller pieces of wood. Which one is going to be more conducive to starting that fire? Well, we know it would be the many smaller pieces because there's greater surface area to volume ratio. There's so much more surface compared to that big log, say, which has a great volume and in comparison a small surface area. So therefore more chemical reactions can take place on the surface, allowing that fire to begin. It would be the many little pieces of wood. So cells are also conducive to doing what they need to do, being small because they have a greater surface area to volume ratio. Why is that important? Well, we want surface area to allow things to come in and to allow things to get out, like important things such as oxygen to come in easily and wastes such as carbon dioxide to be able to diffuse out. It's also helpful having a smaller volume because then if we look at this image, the oxygen doesn't have to travel so far to get into the cell, and same with carbon dioxide. So if that is the case, you might wonder, well, why aren't cells infinitely small? And it's because there's this balance here. Cells have to be able to fit their machinery, all the organelles, all their parts within the cell. So there's kind of like this sweet spot whereby for prokaryotes and eukaryotes, which we will talk about next week, we can see that they are similar in size, like all prokaryotes and all eukaryotes, they have a similar size kind of window, and it's because they have this optimal range of surface area to volume ratio. We're going to do an experiment on this in class as well to kind of see this in real life action. So that is it as far as content goes. And homework is to read about what we have just spoken about. So that was 1.3 to 1.6 of the text. Of course, be adding notes and key terms to your glossary, whether that's a page at the back of your book or just as you go with your class notes, or it might even be actual flip cards that you're creating, physical flip cards, or using something like Quizlet to do that. And then I want you to research something that links back more so to last week, and it's this idea of hypertonics, hypotonics, and isotonics solutions and how they have different roles. So in hospital, you might have heard that, or maybe you've had one yourself, being hooked up to an IV, an intravenous, that means like straight into the blood supply solution. I want you to research what kind of solution do we use? Is it pure distilled water? Or does it have something else in it that changes the concentration of the solution? Find out what it is and explain why that's so important and how actually having the wrong solution going into our body can be so disastrous. So bring that to class on the last lesson of week 3, where we can have a discussion about that. If you'd like more support, there's an awesome video by the Amoeba sisters on everything that we spoke about on cell transport, and I'll put the link in the description of this video. So I'll pause the video here and you can jot that down. a couple of just animations to help us visualize what we spoke on. So when we are going against the concentration gradient, that means like going uphill from low concentration where there's not much of it, and we want to put whatever it is where there is more of it, it's not going to just naturally happen through the process of diffusion. So we need energy, and that is in the form of adenosine triphosphate, or ATP. It's the currency of our body. Active transport. We have active transport, like we just spoke about. Then we have bulk transport, and an example being endocytosis. And this is where endo meaning it's coming in, so something fuses with the cell membrane, creates a vesicle around it, and then it comes into the cell. The opposite would be exocytosis. But that's all active transport. Something that does not require energy is passive transport. And an example of this is osmosis, that is the movement of water across a semi-permeable membrane. Another kind of broader example of that is just the process of diffusion. So this might be oxygen coming into the cell, where it's still going from a high to a low concentration, but just needs a bit of help. We have facilitated diffusion where proteins can play a role in that. And here it looks like we have a protein channel. This could be glucose coming into the cell. So one more. An example of active transport is the sodium-potassium exchange pump, whereby ATP binds to the protein, and then 3 sodium ions go from intracellular to extracellular environment outside, and in exchange for 2 potassium ions that then move inside the cell against their concentration gradient. Lastly, just a couple of other ways to think about this. Here we have, I might just move myself here. 2 star-crossed solids. Where art thou, Romeo? I'm stuck behind this semi-permeable membrane. Then we have the semi-permeable membrane, which is putting the passive back into passive transport. I like this one the most though. So here we have a wall with transport proteins inside and outside, and if you get it, it's because it's a cell wall. I hope that this lesson has been informative, and in conjunction with your readings and everything that we do in class, that you guys can really get a firm understanding and grasp on everything that we spoke about. So God bless you guys, and I'll see you in class.